All right, so today we are completing our discussion of how a skeletal muscle contracts. And if you remember, a quick overview of what we've mentioned last time. We first discussed the structure of the contractile unit of the skeletal muscle cell. And if you remember, the contractile units of skeletal muscle cells, those are my sarcomeres. And remember, your muscle fiber, which is the muscle cell, is formed of sarcomeres, which are lying adjacent to one another throughout the length of the muscle fiber. So you've got many sarcomeres forming each of the muscle cells, each of the muscle fibers. If you remember the structure of the sarcomere was formed of alternating thick and thin filaments. So you've got thick filaments which are alternating with thin filaments, as you see on here. If you remember, the thick filaments are going to be attached to one another by what we call the M line. All those thick filaments together will appear under the microscope as this striation, as one of those striations along the length of my muscle fiber. All of this together. So the line formed by the thick filaments appears darker in color under the microscope. So if I'm looking at the whole thing, all those together will appear as one striation along the length of my muscle fiber. So another one means this is going to be another sarcomere, and another one means another sarcomere, another sarcomere. So each of those striations mean simply I have part of the sarcomere which is formed of thick filaments lying over on top of one another. Alternating again with those thick filaments, we've got those thin filaments. And if you remember, thin filaments are going to be attached at the periphery of my sarcomere by something that we've called the Z line or Z disc. So again, thick filaments are attached to one another at the middle by the M line. Thin filaments are attached to one another by the Z disc. Simply speaking, what is the sarcomere? This is the distance between two successive Z discs. All this is what we consider the sarcomere, which is the contractile unit of my skeletal muscle fiber. The zone that is formed only of thick filaments without having thin filaments alternating with, so all this part on here is going to be my H zone. So all this part in the middle of the thick filaments, the dark band of the thick filaments, we call this as my H zone. So the part occupied by the thick filaments is what we call here the dark band or the A band. And on both sides of the A band, we've got parts that only going to be consisting of thin filaments. without thick filaments. So those parts on here are my light bands or my I bands. So again, if I'm drawing here the sarcomere, the structure of the sarcomere is going to be formed of thick filaments. Lying on top of one another. Like this. Alternating with those thick filaments, you're going to have thin filaments. All right. Attaching the thick filaments to one another, like this. This is going to be my M line. Attaching the thin filaments together. Those going to be my Z discs. All the thick filaments will appear darker in color under the microscope. This will appear as a striation. So we call those all together. This is my dark band or my A band. And this is a part formed by thick filaments on top of one another. On both sides of the A band, you've got parts which will appear lighter in color. They consist only of thin filaments. So we're going to call those are my light bands or my I band. In the middle, you've got parts of the A band which are not covered with the thin filaments. Parts of the thick filaments that are not covered with thin filaments. We call this is my H zone. What is the structure of those thick and thin filaments? Thick filaments are formed by protein subunits called the myosin. On the other hand, my thin filaments are going to be formed of 3 different types of protein subunits, but the main component that builds up my thin filaments are going to be my protein subunits, which are called the actin. So again, thick filaments are formed of myosin. Thin filaments are going to be formed by mainly actin. This is the main component that builds up my thin filaments. What do we call the whole thing? The distance between two successive Z discs that are attaching my thin filaments together. Call this is my sarcomere, which is the contractile unit of the muscle fiber. So one sarcomere appears as a dark line. On both sides of it, you're going to have light colored parts. And then another one, and then another one, and then another one, then another one. So it's all throughout the length of my skeletal muscle cell or muscle fiber. Same, muscle cell is called muscle fiber. And if you remember, muscle fibers are going to be attached to one another by connective tissue. And all those muscle fibers together will build up my whole muscle. Let's say this is my biceps. So I'm formed from inside by bundles of muscle fibers. And again, inside each of them, you're going to have sarcomeres lying adjacent to one another throughout the length of the muscle cell. Is this clear? Is this clear so far? Anybody has questions? Again, this is one unit of all those units that are forming my muscle cell or muscle fiber. All right, so remember, how did we start the voluntary skeletal muscle movement? Here we're concerned with voluntary muscle movement. We are concerned with skeletal muscles. So skeletal muscles, as you remember, those are under voluntary control. So where do I initiate the signal from? I initiate the signal from my cerebral cortex, which is the outer part of my cerebrum, the largest part of your brain. I've got a nerve cells that will be sending down the electrical signal along its length, down from the brain through the brain stem and then to the spinal cord. And we're looking here at a transverse section of the spinal cord. This is my spinal cord on here. This is where you're going to be connecting this nerve cell to another nerve cell that exits the spinal cord through a nerve. So what kind of signals are you conducting throughout the length of the nerve cells? You're conducting electrical signal. And we did discuss a little about what I mean by an electrical signal that travels along the plasma membrane of the muscle cell and nerve cells last time. We're going to discuss this in details in a later chapter. This electrical signal will reach the terminal end of my nerve cells, which are directly connected to my muscle cells, muscle fibers. So if I'm drawing a single nerve cell like this is how a nerve cell gonna look like. It has a cell body. It has short tapering processes. And it has a long process. We call this the axon. The terminal end of the axon gonna have processes coming out, and in the terminal end of each one of those processes, you're gonna have what we call the terminal button or terminal node. This is where you're gonna be releasing, this time, not an electrical signal, but a chemical signal. So I've got an electrical impulse. traveling down in here through the nerve cell. This electrical signal is traveling down through the nerve cell along the length of the axon to the terminal ends of the axon. And this electrical signal is what's going to be stimulating the release of the chemical signal to be released from those terminal nodes or terminal boutons. So again, where do I get this electrical signal? It was initiated in the cerebral cortex. So you've got an electrical signal traveling down in here through this first nerve cell, and then we'll send the signal to another nerve cell, and this other nerve cell coming out of the spinal cord gonna be conducting the signal to reach my muscle cells. Here we are magnifying the junction between the muscle cell and the nerve cell. This point. We are looking at one of the terminal ends of the nerve cell and one of the muscle cells they get in contact with. So this is my nerve cells. This is the terminal bouton of the nerve cell. I've got an electrical signal down in here, and this is one magnified terminal bouton. When I get the signal all the way to the terminal boutons on here, and this is one of them, what's gonna happen? The electrical signal gonna cause the opening of a voltage gated calcium channel. So I open a channel that allows calcium to travel to the inside of the terminal end of the nerve cell. This calcium influx is gonna be stimulating vesicles that are kept in the terminal ends to fuse with the cell membrane. And if you remember, we called this as an exocytosis. If you remember what's exocytosis, it's a type of active membrane transport in which you're gonna have vesicles, and those vesicles will fuse with the cell membrane. And as they fuse with the cell membrane, they will be releasing their contents to the outside. This is what's gonna happen. What did stimulate those vesicles inside my terminal end of the nerve cell to fuse with the cell membrane and release their chemicals to the outside on here? In the junction between the nerve cell and the muscle cell is the entrance of calcium. So you've got calcium in, and as you get the calcium in, this gonna be stimulating those vesicles to release their contents at the junction between the nerve cell and this one is my muscle cell. If you remember, what do we call the muscle cell membrane, skeletal muscle cell membrane? This is my sarcolemma. So now I've got the chemical here, the neurotransmitter. We call the chemicals that will be released from the nerve cells are my neurotransmitters. And in this specific example, the neurotransmitter is called acetylcholine. This is a chemical that gets released once you've got a signal down to the terminal end of the nerve cell. What's going to happen when you get the acetylcholine released at the junction between the nerve cell and the muscle cell? Acetylcholine has its receptors on the sarcolemma. This is my sarcolemma here. This is the plasma membrane of the muscle cell. On this outside surface of the sarcolemma, you've got receptors for the acetylcholine. So once the acetylcholine gets released, it will bind to its receptors. Those receptors are associated with ion channels. So if you have the acetylcholine, what's going to happen if I'm magnifying here? This is my acetylcholine. This is a receptor for the acetylcholine. And associated with this receptor, you've got an ion channel. Normally, it's kept closed. Once you bind the acetylcholine to its receptor, this will cause a change in the shape of the receptor as well as the associated ion channel. This allows you to open the channel. So again, what's gonna allow you to open the channel is the attachment between the acetylcholine and its receptor located on the sarcolemma, which is the plasma membrane of the muscle cell. So you've attached the acetylcholine to the receptor, you've opened the channels. Those channels will allow sodium to travel to the inside of the cell. And if you remember, a sodium ion is positively charged. What happens when I open the channel for the sodium? Sodium travels from its high concentration gradient outside of the cell to its lower concentration gradient inside of the cell. High concentration to low concentration. And as a result, what's going to happen? I gain more positively charged ions on the inside of the cell membrane. So now the inside of the cell membrane will become more positively charged. And if you remember, what is the difference between having an electrical signal and not having an electrical signal traveling through the plasma membrane? Remember, how do you conduct the signals in your electronic devices? You either gonna have a signal, an electrical signal, which is coded as 1, or you don't have an electrical signal, which is coded as a 0. And your computer or your tablet or whatever you're using will be getting millions and millions of those signals. Either you have a signal and then you don't have, or you have signal followed by another signal followed by another signal, and then you don't have signal for a while, and then you've got a signal, and so on. So you have a sequences of zeros and ones, and according to the code you're going to be receiving, you're going to be able to see something on your screen by processing this signal, those signals that carry the information. And the processor speed is going to be determined according to how fast I'm able to retrieve the code and decode it into another signal that can be perceived by the screen, for example, or the speakers. All right, so the same concept on here in the nervous system, if you remember from last time. If I don't have a signal, I'm not carrying a signal through my nerve cell or my muscle cell, it's a 0. How is a 0 coded in your nervous system? Inside of the cell is negatively charged, outside of the cell is more positively charged. This is a very simple way to look at how do I conduct the signal. What if the inside becomes more positively charged than the outside, which will become more negatively charged? Like in here, I did allow what ion to travel. When I get the acetylcholine attached to its receptor, I'm allowing the sodium to travel from outside of the cell to the inside of the cell membrane. So the inside of the cell membrane will become more positively charged. So now I've got a signal. So again, what does it mean for me to not have a signal, to have a zero? I'm not conducting a signal. It means that the inside of the cell membrane is more negatively charged and the outside of the cell membrane is more positively charged. Conducting a signal, an electrical signal, means what for you? Means inside is more positively charged compared to the outside, which is more negatively charged. This is just a simplified way to understand how do you conduct electrical signals through the nervous and muscular system. We're going to discuss more details about this in the next chapters. So again, what happened so far? I've created an electrical signal from my cerebral cortex. It travels down along the nerve cell through the brainstem and then the spinal cord. This is where you're going to be conducting the signal from this nerve cell to another nerve cell. The other nerve cell will be carrying the electrical signal, still electricity, to its terminal ends, where you're going to have the junction between the nerve cell and the muscle cells. What happens in the terminal ends of the nerve cell? Having an electrical signal conducted along the length of the nerve cells, this allows you to open calcium channels. And we call those calcium channels, if you remember, voltage-gated calcium channels. Again, why do I call them voltage-gated calcium channels? Because normally they are gated, they are closed. And in order for them to open, they need electricity. So we call those are voltage gated and they are allowing calcium to travel. So it's going to be a calcium channel. So we call those channels that get open by the effect of electrical impulse traveling down along the length of the nerve cell. It's called voltage gated calcium channel. Again, what's going to be the effect of this calcium when it travels to the inside of the cell? Calcium mobilizes the vesicles containing the neurotransmitter. And again, what's the name of the neurotransmitter located in the neuromuscular junction that gets released in the neuromuscular junctions? This is my acetylcholine. Acetylcholine, once it gets released by exocytosis from the nerve cell, it binds to its receptor. Associated with the receptor of the acetylcholine, once the acetylcholine binds to its receptor, you're gonna be able to open sodium channels. When I open the sodium channels, what's gonna happen to the sodium? Sodium will move to the inside of the cell, making the inside to be more positively charged. So now I've created another electrical signal traveling this time not along the nerve cell, but along my muscle cell membrane. Any problem? Any problem so far? Questions? All right, so now I have an electrical signal, but this time it's traveling along my sarcolemma, the plasma membrane of my muscle cell. In the sarcolemma, we've got parts of the membrane that are traveling to the inside of the cell, like this. We call those are my T-tubules. So now I have the signal that was traveling along the sarcolemma, along the plasma membrane of the muscle cell. Now it travels down through my T tubules. In the T tubules, you're going to have another voltage-gated calcium channel. Remember, voltage-gated calcium channels would open when you've got an electrical impulse. So this electrical impulse gonna open this calcium channel, allowing calcium to pass to the inside, this time of my skeletal muscle, but not directly to the cytoplasm of the cell. It's gonna be allowing the calcium to travel to flattened tubules located within the skeletal muscle cell. This is my smooth endoplasmic reticulum of the skeletal muscle. This is what we call my sarcoplasmic reticulum. So this calcium doesn't pass from outside of the cell to the inside of the cytoplasm of the cell, but it travels to the inside of those flattened tubules of my endoplasmic reticulum. We've got calcium here from outside under the effect of the electrical impulse that you've created earlier due to the influx of the sodium. As the calcium passes to the inside of the endoplasmic reticulum, it stimulates my endoplasmic reticulum to release the stored calcium on the inside. If you remember when we studied the cell back in chapter 3, when we studied the organelles of the cells, we've mentioned the function of the ribosome was to produce proteins. We mentioned that the rough endoplasmic reticulum function was to allow the membrane-bound ribosomes to produce their proteins. We've discussed the function of the Golgi apparatus, and this was the location where you've got the modification, concentration, and packaging of those proteins. And if you remember, when we discussed the function of the smooth AR, smooth endoplasmic reticulum, remember the function was different between the different organ cells. So for example, in the liver, it was responsible for the detoxification of the toxins, the storage of the fat. But here, what was the function of the smooth endoplasmic reticulum in the muscle cells? It was to store calcium. So if I'm storing calcium inside, why would I need calcium from outside? Calcium, as it travels to the inside of the endoplasmic reticulum, it enhances the smooth ER to release the stored calcium out in the cytoplasm. So I've got a lot of calcium now released from the endoplasmic reticulum out in the cytoplasm of the skeletal muscle cell. So we call this mechanism, it's a calcium-induced calcium release. So what this means, that this calcium that has been released inside the cytoplasm of the cell this calcium on here was released under the effect of another calcium coming from outside. So the induction of the calcium release is taking place by another calcium that is coming from outside. We call this as my calcium-induced calcium release. Did I have any contraction of the muscle cell right now, so far? Did you get any muscle contraction? Did I mention about anything about shortening the length of the muscle cell? No, not yet. Not yet. All this gonna be taking place before the muscle starts to contract. All this. So what I started with here, I initiated an electrical signal in my brain, and I ended up having calcium released in the cytoplasm of the skeletal muscle cell. All right? How is this calcium gonna allow you to contract your muscle cell? This takes us back to the internal structure of the muscle cell. If you remember, what are the contractile units of the skeletal muscle cells? Muscle fibers. Those are called I can't see any answers. What are the contractile units that build up my skeletal muscle fibers? One answer. One answer. Contractile unit of the skeletal muscle cells. Here we go. Contractile unit of skeletal muscle cells is my sarcomere, exactly. Sarcomere. Sarcomere is the contractile unit of the skeletal muscle cell. All right, so how's the sarcomere gonna be affected by the presence of calcium? So you've got calcium inside your skeletal muscle cell like this, out in the cytoplasm. Again, where did I get this calcium from? From my smooth ER. Calcium-induced calcium release. So how is this calcium gonna allow you to contract your muscle? So this needs us to magnify the connection between a thick filament and a thin filament. Again, a thick filament is formed of myosin subunits like this. And thin filaments are going to be formed mainly by actin subunits. Also, in the structure of my thin filaments, I have two other subunits other than the actin. Those are my troponin and attached to the troponin, you've got long arms like this. This is called my tropomyosin. So what's the effect of the calcium? As the calcium increases out in the cytoplasm, it has been released from the sarcoplasmic reticulum. The calcium binds here to the troponin. And as it binds to the troponin changes its shape and will be moving away the tropomyosin. So rather than having the tropomyosin in here, it's gonna be moving down like this. And I don't have this one anymore. So what did allow the tropomyosin to move from its place is the attachment between the calcium and the troponin. Once the tropomyosin moves away from its place, this will expose binding sites on the actin subunits, like this. Those binding sites are going to be allowing the myosin to bind to the actin, forming cross bridge. And as the myosin forms the cross bridges will be bending, pulling the actin to move towards the M line. So what's going to happen in here? This actin, let's say this is actin number 1, it's going to be moving from its place towards my M line. This is actin 1. And this one is going to be actin 2. And this one is going to be actin 3. So what happened in here, I did pull the whole filament down along the length of my thick filaments. So what happens again? Thick filaments are forming cross bridges that will bind to the actin subunits, pulling them towards the M line. So what's going to happen? The actin units are going to be moving along the length of my thick filament. So this is going to be my actin 1, this is going to be 2, this is going to be 3. This is my tropomyosin here. What happens again? The myosin subunits will be binding to the actin, pulling the actin subunits to move along the length of the thick filaments towards my M line. Remember what was the M line? M line was the filament that attaches my thick filaments together. This is my M line on here. So what's going to happen to the length of the sarcomere? If you notice on here, those are my thick filaments, and those thick filaments are formed mainly by actin, so they are pulled towards the M line from both sides. As you pull them from both sides, what's going to happen to the length of my sarcomere? This was the length of the sarcomere before I contract. Now this is the length of the sarcomere after I contract. So you're going to be shortening the length of the sarcomere. I am reducing the length of the sarcomere, and if I did reduce the length of each one of the sarcomeres, remember the whole muscle cell is formed of sarcomeres lying adjacent to one another. So if you shorten each and every one of them, so if this is the distance of the sarcomere, is the length of the sarcomeres, this is another sarcomere, this is another sarcomere, this is another one. If I shorten each and every one of them, what's going to happen to the length of the whole muscle cell? Shortens. And if you shorten each and every muscle cell, what's going to happen to the length of the whole muscle? Becomes shorter. And as the muscle becomes shorter, it's going to be able to perform its action. Let's say my biceps brachii on here is attached to my radius of the forearm, like this. And it has two heads, a short head and a long head, coming from the scapula. So when I shorten the length of the biceps brachii. What's going to be the effect? You're going to be pulling the radius to move towards your scapula. This allows flexion of the forearm or flexion of the elbow, as what we've learned in the previous chapter. Anybody is confused here? Anybody is here? So let's watch this animation here together to make life easier. All right, so this animation is showing how the myosin subunit is going to be pulling the actin subunits, causing the shortening of the length of the sarcomere, and as a result, shortening the length of the muscle cells. And by the end, you're going to have a shortening of the whole muscle. So let's start on here.
This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate.
So you notice on here, I'm using energy to form the cross bridge coming from the myosin to bind to the binding sites on the actin subunits. So you need to break down the bond between an ADP and an inorganic phosphate. I need to consume to break down ATP to generate the energy needed for me to form this cross bridges to allow the myosin to bind to the actin. Also notice that in order for you to detach to bind to the next actin, you're gonna need another ATP.
This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber.
What I mean by depolarization of the membrane is creating another electrical signal. And remember, the other electrical signal here is allowed by the influx of sodium ions to the inside of the muscle cell. So this depolarization is a creation of an electrical signal, this time on the muscle cell membrane. And this is due to the attachment of the acetylcholine to its receptor that allowed you to bind on here, that allowed you to open the sodium channels for the cell membrane to become more positively charged on the inside compared to the outside. This is what we call depolarization. This is my electrical signal in the skeletal muscle cell membrane.
The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body.
So I want you to check on here's this one.
To the M line.
If I'm contracting each one of the sarcomere, if I'm shortening each one of the sarcomere, check on here what's going to happen to the length of the muscle.
Myofibrils contract, the entire muscle.
Shortening each one of those will allow the whole muscle fiber to shorten. And if you shorten each one of those muscle fibers, you would be able to contract the muscle, which is essentially formed by muscle fibers. Any questions? Questions? All right, so today's activity is going to be to summarize what you've learned from this video. So where to can find this video. So this video recording, please check on YouTube, muscle contraction process HD animation. So the activity for today is going to be to write a summary of what you did understand from this video recording. It's going to be called the muscle contraction process HD animation. Look it up on YouTube and it's going to be the first thing to pop up. All right. Clear. This is our first activity for today, to write a summary of what you've understood from this video recording. All right, again, it's called the muscle contraction process HD animation, and you can look it up on YouTube. Any questions? right, so again, let's go over this part together from your handout. Remember, a muscle cell is formed by, or a muscle is formed by muscle fibers. And a cluster of muscle fibers is gonna have those myofilaments, those thick and thin filaments on the inside. Those are what we call the myofilaments. What is the sarcomere? Sarcomere is the smallest contractile unit, the functional unit of a muscle fiber. It is the region of the myofibril between the two successive Z discs. What again are the Z discs? Those are the attachments of the thin filaments to one another. Skeletal muscle is going to be formed of alternating thick and thin myofilaments that are made of contractile proteins. Thick filaments are going to be running the entire length of the A band, or what we call the dark band. This is what appears as a striation under the microscope. Thin filaments will run the length of the I band and part way into the A band. As you remember on here, thin filaments gonna be running along the I band, which is the light band of the sarcomere, and not all the A band, parts of the A band as well. Z discs, those are cone-shaped sheets of proteins that will be anchoring the thin filaments and connecting the myofibrils to one another. What is the H zone? The H zone is the part that has lighter color in the mid region where the filaments do not overlap. I'm formed only of thick filaments. If you notice on here, what is the H zone? H zone is this region where I have only thick filaments not alternating with thin, only thick filaments not alternating with thin filaments. This is my H zone. The M line is a line of proteins that will be attaching the myosin filaments together. So looking closer at the structure of the sarcomere, again, the sarcomere is the distance between two successive Z discs. It's formed of alternating thick and thin filaments. Thick filaments are formed by myosin subunits attached to one another at the center of the sarcomere by the M line. Alternating with the thick filaments, you've got your thin filaments, which are mainly formed by the actin. Thick filaments are running the entire length of the A band. So all this is my A band, the dark band, compared to the thin filaments are running the length of the I band or the light band, as well as part of the A band. Again, sarcomeres lying adjacent to one another are going to be building up my muscle fiber or muscle cell. Z discs hold what together? Thick filaments, sarcomere, I band, thin filaments. Z discs are holding together the thin filaments, exactly, thin filaments. Thick filaments are composed of protein subunits called the myosin. The myosin heads form cross bridges with actin of the thin filaments, and they have binding sites for the ATP to get attached. Thin filaments are composed of 3 protein subunits, but the most important and most abundant of them is going to be the actin subunits. 2 other regulatory proteins that are associated with the actin that share in the formation of the thin filaments, those are the tropomyosin and the tropon. Actin has the binding sites for the myosin heads. So looking closer at the structure on here of the thick filaments formed of myosin subunits. They have myosin heads that will allow them to bind to the actin. Compared to the thin filaments, they are mainly formed of actin subunits. On each one of them, you're going to have the binding site for the myosin head. Because this is the myosin binding site on the actin subunits. Associated with those actin subunits forming my thin filaments, you've got the regulatory protein subunits. Those includes the troponin, and those here, those filaments are called my tropomyosin. So again, thick filaments are formed of the myosin subunits, which will have the myosin heads. Each one of the myosin heads is going to have the actin binding sites, and also it has a binding sites for the ATP, for the energy molecule for it to bind, and causes its extension for it to bind to the actin. Skeletal muscle fibers. Remember, what is the sarcoplasmic reticulum? Sarcoplasmic reticulum, this is a smooth endoplasmic reticulum of the skeletal muscle cell. It's a network of smooth endoplasmic reticulum tubules surrounding each myofibra. The function here will be to regulate the levels of calcium in the cytoplasm of the skeletal muscle cell. So remember, what did allow the muscle to contract is the release of the calcium from the sarcoplasmic reticulum. So where did you get this calcium from? Again, it's from the inside of those flattened tubules. T-tubules are continuous with the sarcolemma, which is the plasma membrane of the muscle cell, as we've seen on here. It's a continuation of the sarcolemma that travels to the inside of the muscle cell, allowing the conduction of the signal, electrical signal, to reach my sarcoplasmic reticulum, where you are storing the calcium. Those T-tubules are going to be located at the inside of the muscle cell between the A and I bands. This allows the electrical stimulus to be delivered deeper into the cell. We're looking here. This is one sarcomere, if you notice on here. It has the A band and it has the I band. This is my I band on here. This is my A band. Remember, A band is the one that consists of thick filaments running throughout its length. I band, it's only going to be formed of thin filaments. Remember, where do I get those T tubules traveling to the inside of the cells? They are traveling as a junction between the A band and the I band to carry the electrical signals deeper into the sarcoplasm. Muscle contraction is going to be due to sliding of the myofilaments. In a relaxed muscle state, thin filaments and thick filaments will overlap only slightly. During the muscle contraction, the myosin heads will bind to the actin and then will detach from the actin, will bind to the next actin, and this allows you to propel the thin filaments along the length of the thick filament. So if I have the thick filament again, this is just one actin. So this one is going to be attaching this actin like this. It's going to be pulling the actin towards the M line. So it's going to be moving to this side. And then we'll detach from it for the next myosin to form a cross bridge and bind to it and pull it again. So now the actin did move like this. This allows what to take place? This allows shortening the length of the sarcomere, which is the distance between the two successive Z discs. So the length of the sarcomere is going to be shortened if I am pulling here those thin filaments towards the M line. So if I am putting the thin filaments towards the M line, what do you think is going to happen to the H zone? Do I increase the length of the H zone or it will shrink and disappears? What do you think? What was the H zone by definition? What was the H zone by definition? It is the part occupied only by the thick filaments, which are not overlapped with thin filaments. So when you are pulling those thin filaments towards the M line, so rather than having them like this on here, stopping at this point, they will keep moving like this. So what's going to happen when you pull those actin towards the M line? There will be no region where you have only thick filaments, not alternating with thin filaments. So the H zone will disappear. I will shrink the H zone and the H zone disappears. If I look in here, this is my Z disc. This is my Z disc on here. This is another one. So what do we call the distance between the two successive Z discs? This is my sarcomere, the contractile unit of the skeletal muscle cell. Remember, sarcomere has A band occupied by thick filaments. And I have the light colored part, light band. This is called my I band. And remember, in the middle of the A band, you've got a zone where you're going to have only thick filaments, not alternating with thin filaments. So this is what we've called the H zone. So now I'm pulling those Z discs towards one another, like this. Sliding those thin filaments towards my M line. So what happens? H zone disappears. And the two Z discs are drawn towards one another. So the length of the sarcomere shortens. So compare those two to one another. Look at those two diagrams, compare them to one another. Which one is shorter in length? A or B? Which one has a shorter sarcomere? Again, my question, which one has a shorter sarcomere? What is the sarcomere again? The distance between those Z lines or Z discs. Which one is shorter in length? A or B? B. What was this region formed of only thick filaments and not overlapped with thin filaments in the middle of the A band? What do we call the part in the middle of the A band that is formed only of thick filaments, not overlapped with thin filaments? What do we call the region on here? Has only thick filaments, not overlapped with thin filaments. This region on here, what did we call it? H zone, exactly. H zone. So what happened to this H zone? You still have it in here? When you contract the muscle, you still have an H zone? Yes or no? No H zone. H zone, where did it go? Disappears. So you see on here, I've got thick filament like this. This is my thin filament. The part that has thick filaments not overlapped with thin filaments, this is my H zone. When I pull those actin subunits like this, so all the thick filaments are going to be overlapped with thin filaments. So no H zone will remain when I contract the sarcomere. Does it make sense? Any question? Any question? All right, so how again muscle contraction takes place? It all started by having a signal coming down from the brain. an electrical signal that travels along two neurons, two nerve cells. And then I will reach the junction between the nerve cell and the muscle cell. At the junction between the nerve cell and the muscle cell, the electrical signal can allow you to open this voltage-gated calcium channel. The junction between the muscle cell and the nerve cell is what we call the synaptic cleft. Synaptic cleft is a gel-filled space located between the terminal end of the nerve cell and the muscle cell. Once I get the electrical signal delivered all the way to the terminal end of the nerve cell, this will enhance the vesicles in my axon terminals to release their contents, and the content here in those nerve endings is going to be my neurotransmitter, which is the acetylcholine. So we can see on here, I've got an electrical signal traveling down along the length of the muscles of the nerve cell, reaching the terminal ends of the neurons. Those terminal ends of the neurons, as they get this electrical signal, this will allow the voltage-gated calcium channels, as you see on here, to open, allowing calcium in. Calcium, as it travels in, it's going to be mobilizing those vesicles and enhancing them to fuse with the cell membrane. As I fuse with the cell membrane, now the neurotransmitter contents of those vesicles going to be released out in the synaptic cleft. This is where the acetylcholine going to be able to reach its receptors located on the sarcomere, on the muscle cell membrane. Once the acetylcholine binds to its receptor, it's going to open channels associated with it to allow the influx of the sodium, to allow the sodium into the cell. Once the sodium passes to the inside of the cell membrane, now I will be creating a state of depolarization. What I mean by depolarization, I simply mean that the inside of the cell is going to be more positively charged compared to the outside, which is going to be more negatively charged. This means I'm conducting an electrical signal now on my muscle cell. This electrical signal going to keep traveling until you get to the T tubules. The electrical impulse passes down into the T tubules that are associated with those flattened tubules on the inside of the cell membrane. What do we call those flattened tubules on the inside of the cell? This is my smooth endoplasmic reticulum of the skeletal muscle cell. This is my sarcoplasmic reticulum. What happens inside the T tubules as you get this electrical signal delivered? The electrical signal traveling down through the T tubules, it's gonna open calcium channels, stimulating the sarcoplasmic reticulum to release its calcium out in the cytoplasm. So now I've got the calcium out in the cytoplasm everywhere. And what was the effect of the calcium that you're going to be releasing outside of the sarcoplasmic The reticulum will be to bind, you see on here, this is my calcium that I did just release from the sarcoplasmic reticulum. It's going to be binding to whom? It's going to be binding to the regulatory protein on the thin filaments, which is my troponin. And as it binds to the troponin changes its shape, pulling away another regulatory protein. This is my tropomyosin. Tropomyosin, as it moves from its place, this allows you to expose the binding sites on the actin subunits. Those are the binding sites on the actin subunits. Once you get them exposed, who is waiting for them to get exposed? Your myosin heads. The myosin head extends to bind to the binding site on the actin subunit, pulling the actin filament toward the M line. So I'm extending the arm, binding to it, and will bend, pulling the actin towards the M line. So what you're going to do next, as you pull the actin, remember, in order for you to perform this action, to extend the arm, you need to get ATP to bind, and once you bind to the actin, you pull it towards the M line, and then in order for you to detach, you need another ATP to detach for you either to relax or for you to keep contracting, for you to bind to the next one. You bind, then bend, then detach, then bind, bend, and then detach, bind, bend, detach, bind, bend, detach, bind, bend, detach. It's a cycle that takes place until you reach the degree of contraction that allows you to perform the action that you want. All right, so can you answer this question here for me? What neurotransmitter is required for muscle contraction to take place? What was the name of the neurotransmitter? Neurotransmitter. What is the neurotransmitter? This is the chemical that gets released from the nerve cell. What is the chemical that gets released from the nerve cell? It's my acetylcholine. Calcium is needed for the muscle to contract, but it's not the neurotransmitter. It's not released from the nerve cell. Again. If this is my nerve cell on here, it gets an electrical signal traveling down. This electrical signal allows calcium to get in the cell, but this calcium is needed for you to release your neurotransmitter out. The neurotransmitter is the chemical release from the nerve cell that will be binding to the muscle cell. So again, what do we call the neurotransmitter that gets released from the nerve cell to bind to the muscle cell? It's my acetylcholine. Is this clear? Is this clear? So again, calcium is essential for the muscle to contract, but it's not the neurotransmitter. Neurotransmitters are chemicals that get released from the nerve cell. Do you release calcium from the nerve cell, you don't release calcium from the nerve cell. Calcium enters the nerve cell for you to release acetylcholine. So we can't consider calcium as a neurotransmitter. Calcium is not a neurotransmitter. What is the neurotransmitter again in here? It's gonna be my acetylcholine. Questions? All right, great. So we can have a 10 minutes break. We come back, discuss more about the physiology of the skeletal muscle contraction. We have a break for 10, come back, discuss more about skeletal muscle contraction.